if you amp up photosynthetic rate, then the nutrient concentration that's in those plants comes along for the ride. It's an automatic. And that gets really exciting because now we're talking about stretching forage supplies. So even though the biomass may not have significantly increased, but because your nutrient density really increased, it's like your forage is being stretched.
The primary goal of grazing, or really any farming endeavor, is to efficiently and profitably convert light energy from the sun into food. Many graziers think of themselves as grass farmers, harvesting sunlight and converting it through forage into animal products. In this episode of Voices from the Field, NCAT Sustainable Agriculture Specialists Lee Rinehart and Justin Duncan talk specifically about photosynthesis and whether it can actually be managed. And if so, how? This is a different spin on grazing management if indeed it's possible to optimize photosynthetic activity and get more nutrient-dense forage for longer in the grazing season. Let's listen.
Hello and welcome to the ATTRA Voices from the Field podcast. For more than 35 years, the National Center for Appropriate Technologies ATTRA Sustainable Agriculture Program has been helping farmers and ranchers grow nutritious food and operate successful farm and ranch businesses, all the while reducing high-cost inputs. I'm NCAT Agriculture Specialist Lee Rinehart, and in this episode of the podcast, I'm going to be talking with NCAT Regenerative Grazing Specialist Justin Morris. We're going to be talking about photosynthesis, carbon dioxide, and water. This is kind of a fascinating topic that we've chosen here because it's something that Justin and I have been kind of talking about, kind of informally, for a while, and we're gonna have a kind of bring that to the world today with some of our thoughts and see what we can come up with. You know, we can't manage sunlight; that's a given. You know, the three things that you need for plant sugar development, we can't manage sunlight, and we're learning more about how to manage water. But in today's podcast, we're going to consider if we can manage photosynthesis to facilitate optimal plant growth. And it should be a really interesting conversation. So Justin, hi, and welcome to the podcast today.
Yes. Thank you so much for having me. It's great to be here and kind of be visiting with you during this podcast and kind of dive into some of these topics.
Mm-hmm.
We kind of touched on these a little bit in our advanced grazing webinar or session 2.
Mm-hmm. Yep.
So we'll get to spend a little bit more time talking about some of these nuances that we didn't get to cover as much in depth when we did the advanced grazing webinar.
Yeah, well, you know, and those webinars served as fodder for an awful lot of concepts that we've brought to blogs and podcasts just to kind of do a bit of a deeper dive. And I know that, you know, you've been doing a lot of reading and looking into some of the old research and such, and you got some really good ideas. I'm looking forward to having that discussion today. You know, we know that our primary goal as grazers, or really with any farming endeavor, is to efficiently and profitably convert that sunlight energy into food, right? Many grazers,
exactly.
Many grazers think of themselves as grass farmers, right? They harvest sunlight and they convert it through forage into animal products. But this topic that we've been talking about lately about photosynthesis is really interesting, considering can we actually manage it? And if so, how, right? And this is a really different spin on the whole idea of grazing management. If indeed we can optimize photosynthetic activity and get more nutrient-dense forage for longer in the grazing season, it would be awesome. So, Justin, why don't you set the stage here before we get into the details of managing photosynthesis and catch us up on the relationship between carbon and light, what we're working with here, and kind of what's going on in the plant, you know, and to kind of set the stage.
Great. Thank you so much for that introduction, Lee, and for getting us set up to talk about this. I'm going to go back in time here quite a ways, back to 10th grade high school biology class. And I remember specifically, and that was the first year I ever took biology in school. And one of the main topics that we ended up studying was photosynthesis. And I'm going to kind of assume that pretty much everybody listening here, I mean, we've all heard this term for a long time.
Yeah, we all kind of
And
know the basics of it, right? But
yeah, right. And so, it starts with, plants taking light from the sun and carbon dioxide from the air and water that's absorbed through the plant roots to create the simple sugar glucose. And glucose is the energy currency of the plant. That's the base or the parent compound that's created in plants from photosynthesis. That's the first compound, is glucose. And glucose, it being that core building block, that is what is used to create all the other sugars, amino acids, and other compounds inside the plant.
Right.
So that's the parent compound. So what does glucose look like chemically? Well, it's C6H12O6. So when I say that, I'm saying that there's 6 carbon atoms, there's 12 hydrogen atoms, and there's 6 oxygen atoms. And the hydrogen and the oxygen atoms are held together through the carbon atoms. So the carbon forms like the backbone or the framework for which the hydrogen and oxygen atoms are able to be attached. And that is, for anyone who's delved into chemistry a little bit, if you hear the term organic chemistry, when we say organic, we're not talking about a certification for marketing, but we're actually talking about anything that is carbon based, that's held together with carbon. So the organic chemistry is the science of carbon based chemistry. So that's where your hydrogens, your oxygens, and other elements are tied with carbon. And then the carbon forms either a ring structure or a kind of a linear structure by which all these other elements are able to be attached. So carbon is the backbone. So without the element of carbon, we'd have no glucose and we'd have no life. We wouldn't exist. So carbon is the central element. I think it's number 12 on the periodic table of the elements. And in the photosynthetic process, I'd be a little bit remiss if I didn't also mention that, we not only get sugar development, the creation of glucose from photosynthesis, but we also get, as a byproduct of that, oxygen, O2. So 2 oxygen atoms that are bound together are released from the plant as a byproduct of photosynthesis. So, and oxygen makes up about 21% of the air that we breathe. So that's, critically important for all of us that have to breathe air, is we got to have oxygen. And so that carbon that's in glucose, as well as carbon that is in all the other plant compounds, is really, where does that come from? That comes from CO2, comes from carbon dioxide that is in the air. So that carbon dioxide needs to be in the air in order for photosynthesis to take place. If you shut off carbon dioxide, you shut off photosynthesis. Even though there's ample sunlight, there's ample water, there's other mineral nutrients that are required for photosynthesis, because we talk about sunlight, we talk about water, we talk about carbon dioxide, but there's also important minerals like, manganese, for example, are critically important for photosynthesis to happen. So, but we're just talking just very broadly, just very generally, we're talking about the big 3.
The basic building blocks.
The basic building blocks, sunlight, water, carbon dioxide.
Right.
So when we look at carbon, if you're to take a plant and you were to try to figure out, okay, how much carbon by weight is a plant made up of? Well, it comes out to, roughly somewhere between 50 and 58% by weight. So carbon is far and away the largest constituent in plants, and which then, that makes carbon dioxide absolutely essential for photosynthesis to occur.
Right. So how does this? You have carbon dioxide in the air, and carbon is the basic building block. Explain a little bit then, because we're setting up, right? We're setting up, kind of going over some of these basics here to set up some ideas of how we may be able to manage photosynthesis. And
Yeah.
in order to do that, we have to have a good understanding of how does this carbon dioxide actually get into the plant. So how does the plant take in carbon dioxide from the atmosphere?
Yeah, so that's a great question. If you look at a leaf of a plant, there's a top side to the leaf and there's an underside to it, right? So it's a three-dimensional structure. You've got the leaf edges, you've got leaves of different shapes, but basically, you see, we usually, when we're walking through a field, we see the top side of the leaves. If you turn the leaf under, and if you were to look at it under higher magnification, you'd actually find these little tiny pore spaces on the underside of the leaf. And those pore spaces are called stomates or stomata. And those pore spaces can actually open and close, depending on the environmental conditions that the plant is in. And one of those environmental conditions that really influences whether those stomates open or close is air temperature. So if we're in, if we've got, cool season plants, by and large, once we get above 75 degrees air temperature, maybe 80, then those stomates on those cool season plants start to close. And when they close, that helps to keep water, humidity and stuff inside the plant from leaving because of the high temperatures. So when it gets really hot for a cool season plant, you get into, 85, 90 degrees, really hot weather, your cool season plants are shutting down. They're not photosynthesizing anymore. And the reason why is because those pore spaces not only keep water from going out, but then it also prevents CO2 from going in. So once those pores close, CO2 can't go in, and it's at the leaf area, it's at the leaf where photosynthesis is occurring. And then, you close those pores down and then photosynthesis stops. Now, warm season plants, you take like, Indian grass or big blue stem or switchgrass or Eastern gamma grass or corn. These are all warm season plants, and they have a different photosynthetic pathway called a C4 pathway. And so they can actually continue to photosynthesize at higher temperatures. So when it is 90 degrees out, they can still be making carbohydrates. And so their stomates actually stay open, allowing CO2 to go in and also water vapor to go out. So, the action of these pores, this is the gateway by which CO2 can go into the plant. And then once it gets into the plant, once we have carbon dioxide there, then that process of photosynthesis can then use that carbon dioxide and the water and the plant will actually use the element manganese for water hydrolysis, which then breaks the bond of H2O. So then you get a hydrogen and then an OH separated. And so that's used in the building blocks of creating the glucose molecule. And so that's, kind of in a nutshell, that's how the carbon dioxide's getting in. Then, once that plant makes the building block glucose from that, then glucose is then, the plant will use other enzymatic reactions to then attach other things onto that to convert glucose into other types of sugars or amino acids. And so it will, use photosynthesis to create actually in abundance, actually in excess of what the plant itself actually needs, which is really quite interesting.
Yeah, that's something that I've learned about photosynthesis as well, is that way more sugars and compounds are created by the plant than what it needs. And there's probably a lot of reasons for that. I think one of them could be, just off the top of my head, periods when the plant is slowing down, and during hot periods, the stomata close, and you may still have some compounds in there to allow for metabolic functions to continue or something. But there's other reasons that a plant produces an overabundance of these sugars and compounds. And I wonder if you'd kind of like dig deep into some of those reasons.
Yeah. So one of the main reasons why a plant does this is so that it can lose these compounds. Mm-hmm. And that sounds like really unthrifty of a plant or inefficient. Why would a plant create an excess of these compounds, extra glucose, all these other biochemical compounds, but it's really to lose them. Mm-hmm. And so then we have to ask the question, well, what is the plant losing these compounds to? And the answer then comes in the form of it's losing them to the soil life, the symbiotic organisms, the microscopic bacteria and mycorrhizal fungi that surround the root of that plant. So the plant essentially is building these up to release them through the root system. And there's been several studies out there, like, what percentage does of root exudates, and exudates is a fancy word for, what's being released by the plant root. What percentage of the total carbohydrates that a plant creates is being released. And, I've seen estimates anywhere from, 10% on one end to 40% on another end. And really it's variable because it depends on what's going on in the plant, what the plant needs at the time. So we can't just say it's always 30% because it's not, it varies.
Well, and it probably depends also on the structure of the plant, above and below ground. The more roots, the better, the more surface area. I was doing some reading yesterday, and it seems that not every space on a plant's roots has the ability to exude these carbohydrates out, but most of the plant carbohydrates that are exuded out of the root and into the soil profile occurs at the root tips, because at the root tips, it's more porous at the root tips and allows for this pressure gradient, right? So that you can actually have a flowing of exudates from the roots out into the surrounding soil at the root tips, which tells me it's like, well, then the healthier the plant, the greater the root system, the more roots you're going to have, the more root tips you're going to have.
Right. Yeah, we're talking about increasing surface area here. Mm-hmm. Yeah, and it's basically, you could look at the root tips as basically like a feeding trough.
Right.
For the bacteria to be able to dock up to be able to receive sugars from the plant. And so, and then that kind of begs the question is like, well, why is the plant even doing this? Why is it creating all these sugars just to give them away to bacteria, to mycorrhizal fungi, so then that means, well, okay, so they're feeding them. What are those critters actually doing in return for the plant? Because it's got to be a mutually beneficial relationship.
Right.
So that's where we look at, well, the thing about it is the C bacteria are mobile and they can get into areas of the soil matrix that plant roots cannot get into. I mean, really on a microscopic scale, plant roots are huge. Mm-hmm. They're massive. And so you have a limited surface area of what the plants can actually pull from on its own. Mm-hmm. But one of the things that I stumbled across in doing my research is that plants actually prefer to absorb nutrients in the organic form, meaning that rather than absorbing nutrients in the ionic or inorganic form, like just iron +2 mm-hmm. or calcium +2 just hanging out by itself, it prefers to absorb those nutrients already tied with carbon. And the reason why that is you could kind of think of it like a factory. And I can take no credit for this idea whatsoever, because I heard the similar idea given by another presenter, but then I altered it because I am kind of a tractor geek. So I kind of, made the analogy of a tractor factory, in that you're going to have two different factories. One factory, they cast all the metal, they build all the circuitry, they do everything from scratch under one roof to build a tractor. And then the other factory is one where everything comes assembled as pre-built components. The engine comes down one line already assembled. The transmission comes from another direction already assembled.
Mm-hmm.
The wiring harness has already been pre-assembled. And when you look at the time that it takes to build a tractor, when you got everything pre-assembled, it takes only like 3 hours to build a tractor from the beginning of the production line to the end. And it's like that with plants preferring, just like, it's more efficient to have everything come as pre-assembled components to make a tractor. It's more efficient for a plant to be able to receive nutrients in the organic form, in the form of, sucking in a bacteria. And that's a process called rhizophagy, which we have talked a little bit about in some of our prior webinars, where the plant is actually engulfing bacteria, complete bacteria, stripping the cell membrane off the bacteria, pulling the mineral nutrients that the plant needs, and then spitting that bacteria back out so that that bacteria can go out into the soil matrix and go pick up more minerals to then start the cycle back over again. So there's this marvelous synergy that's going on. And the thing is that the concentration of the root exudates, that if you're to go and look at journal articles to see what the number of compounds that plant root exudates can actually emit, it's hundreds. I mean, it's a vast array. We're not just talking about 4 or 5 different compounds. We're talking about hundreds of compounds that a plant can emit. And really, it changes those compounds in order to provide instructions to the mobile bacteria as well as the fungi of what the plant needs at that time. So the plant knows what it needs, and it can modify its exudation signature, the concentration of the various different compounds, as basically a chemical message to the bacteria saying, I need iron, I need calcium, I need these other things. And then the bacteria recognize that, they can go out and pick up what the plant needs. An amazing symbiosis that's occurring between soil life and the plant root.
Yeah, and it is amazing because these soil microorganisms are living in this habitat within the soil profile, and they're taking root exudates and they're feeding and they're respiring, right?
Yep.
And that brings in another aspect to this whole carbon cycle.
Exactly.
Right. So talk a little bit about, about how carbon is moving through the plant into the microorganisms and then back out into the environment, because it's not just stopping.
No, it's not. Yeah, it's actually a really beautiful cycle. So we talked about, the plant using carbon dioxide as one of the primary ingredients for photosynthesis to create glucose. Then the plant, will take that glucose and then create other compounds, attach other compounds to it, other elements, to be able to create a myriad of biochemical compounds that are then a large percentage of them are then released through the root system to feed soil life. Because you got to remember, bacteria are not autotrophs. Plants are autotrophs. So when I say the term autotroph is an organism that can make its own food. So plants are autotrophs because they can use sun. They can use the power of the sun to be able to create their own food source. Bacteria can't create their own food source. So they rely on the plant to deliver them the sugars and the amino acids that they need to do their job. Well, those sugary substances are full of carbon. That's the number one constituent of the number one element that's in all of that is carbon. So the bacteria, their bodies are using this carbon-based compounds, and many of them are aerobic, meaning that they require oxygen. So they've got to be able to breathe just like we do. But in the byproduct of it, we breathe in oxygen and then we respire out carbon dioxide, right? Carbon dioxide gas. And many of those species do as well. So you've got all these critters down the soil that are breathing in oxygen and then releasing carbon dioxide into the soil matrix. So then that carbon dioxide level, what's happening is As that plant is pumping sugars into the soil, the bacteria are feeding on it. The bacteria are multiplying. So they multiply at an exceedingly fast rate. And now, they're pumping out a lot of CO2. And now where's that CO2 go? Where's it going to go? It's the thing is, carbon dioxide is heavier than air. So if you're to go into a room and if you're to measure carbon dioxide in a room, carbon dioxide is going to be highest at the floor level. And then as you go up in elevation in that room, it gets less and less and less and less because it settles to the bottom.
Mm-hmm.
So and it's the same way outside. So if you were to look at carbon dioxide concentrations in the soil, it could be upwards of 10,000 parts per million. Now, as a comparison, I just did a little bit of searching on the internet, finding that, okay, on average, the CO2 concentration in the Earth's atmosphere is 424 parts per million.
Mm-hmm.
So you compare 424 parts per million to 10,000 parts per million, that's a huge difference. So the soil fills up with all this CO2, and what it does is it builds on itself. It builds on itself and actually pushes as the concentration of CO2 increases in the soil. As long as we've got pore connectivity, and when I say pore, I mean pore, not poor.
Like pore space.
Like pore space.
Right.
Yeah. As long as we've got places for the CO2 to get out and oxygen to go in. And that's one of the probably missing points that isn't talked about that much is, yeah, we want water to get into our soil. But I would say even more importantly for water to get in the soil is gas exchange.
Mm-hmm.
Air that's got 21% oxygen being able to go in and CO2 being produced by soil life being able to get out.
Right.
So as we have this rising concentration gradient of CO2 coming out of the soil, then there's research that goes way back many decades where they looked at what is the concentration of CO2 at the ground surface. And they found that CO2 concentration can be 2 to 4 times that of just the regular air.
Yeah, we're talking a couple of inches off of the soil surface at this point.
Exactly. Yeah. So that proves the point that you can have that CO2 at ground surface is the highest than it is anywhere else in the Earth's atmosphere at the ground surface. And the reason why that has to be is that 422 parts per million is not enough CO2 to help drive photosynthesis long term, to really enhance photosynthesis, to accelerate it, to increase it. We got to have a higher concentration of CO2 for plants. Mm-hmm. And remember, we talked about, probably about 15 minutes ago, we have the stomates on the underside of the leaves. Mm-hmm. So as that CO2 is building up from the ground surface, guess what? Eventually it reaches those bottom leaves, the underside, and then it has a way to go in. So now we are completing a cycle. We're completing a loop. We're getting CO2 to go back into the plant so that we can then continue the photosynthetic process.
Yeah, and that one point right there, I mean, kind of building up from the basics of photosynthesis and what's happening in the plants and root exudation and soil microorganisms and the relationship there. And then also the carbon that is being emitted, and perspired from the soil microorganisms back onto the soil surface completes that loop and really kind of gives us an idea or a place to start whenever we start talking about managing it, right?
Exactly. So this is what kind of caused me to think about this a little bit differently. I was asking myself the question, can we do anything about the amount of sunlight we receive? Well, no, we can't change the number of hours in a day that the sun's going to shine. We can't change things from the aspect of the amount of sunlight that a plant receives by how much leaf area we allow, through our grazing management. If a plant's grazed down really short, it can't receive a whole lot of sunlight. Mm-hmm. If we leave more of a residual, there's more leaf area for the plant to be able to receive sunlight. But we can't change the factor of how many hours in a day is the sun going to shine.
All right.
On the ingredient of water, we can do things to influence that. I mean, if we're in a irrigated environment, we can regulate how much water we're applying. If we're in a rain-fed environment, we can, we can't control exactly how much it's going to be raining and when it's going to be raining, the intensity of that rainfall, but we can change how much cover, how much green cover there's still on the soil surface in our pastures. Because that green cover, if we've got standing, residue or trampled residue, that's going to be important from dissipating the energy force from raindrop impact so that we're not impacting bare soil. And it's going to also change the temperature gradient as well, because, a nice green surface cover is a lot cooler than like bare soil,
Right.
where it's brown. That's going to be a huge difference in temperature. But when we come to carbon dioxide, it wasn't until probably about a year ago I was listening to a webinar given by John Kemp with Advancing Eco Agriculture. And the webinar is entitled How Healthy Plants Create Healthy Soil. And he discussed how to increase photosynthesis. And for those listening, we're going to provide a link to this webinar so that you can go to it and you can watch it yourselves, and it'll be in the resources section once this podcast gets posted. But in that webinar, John said at least 4 things that caught my attention and got me thinking about this as far as pasture management, grazing management. So one of the first things that he says was carbon dioxide is supposed to be delivered from microbial respiration, from bacteria and fungi in the soil, consuming root exudates and other organic matter and releasing carbon dioxide during the growing season. So that was the first thing that caught my attention. Is we want this microbial respiration to take place. We want critters to be exhaling carbon dioxide and lots of it, because it's going to eventually get back to the plant. The second thing that he mentioned is the concentration of carbon dioxide around leaves can drop to 100 parts per million by mid-morning on a warm day, which is insufficient for maintaining high levels of photosynthesis and consequently forage production. So that tends to tell me there is that we've got room to improve. If we're only at 100 parts per million, I mean, that's not even, the Earth's atmosphere level of 424. And we need a much higher level than that to keep photosynthesis driving forward. And then the third thing that he said is that most plants are only operating at 15 to 20% of their genetic potential, and it's possible under the right management and environmental conditions to increase that potential to 40, 50, or even 60%.
Wow.
Now, that I really had to wrap my head around that. I'm like, plants only operating at 15 to 20% of their genetic potential? I mean, that means you still got 80 to 85% left, of room to improve. And so, he was saying, we can get it through the right, management, environmental conditions. We can actually get it significantly higher. We can increase it 2-3 times of what its current level is. And then the last thing that he said is that if photosynthesis doubles or triples, so if we get it from 15 or 20% up to like 50 or 60%, we likely won't see a double or triple in biomass or yield. So, for those of us who are anxious to get tons and tons and gobs of forage, okay, that might be a little bit of a downer, but there's a silver lining to this. And he says the end product of the higher rates of photosynthesis are usually manifest in higher nutrient concentrations along with pest and disease resistance.
So basically just healthier plants.
Healthier plants. And I don't think he would disagree with this point. Depending on the crop that you're growing, and what he was referring to a lot was, typical cash crops, corn, soybeans, wheat, whatever, in a pasture system. And I know you've seen this, and I've seen this in multiple situations where people have really adopted a more recovery-based grazing management, or where they have, really increased they've done, high stock density grazing for certain periods of time. Those people have seen an actual increase in forage production versus not doing those practices, enough so that it has encouraged them to continue doing it in different places of the farm every year. So in a forage production standpoint, I think we could definitely, see an increase in photosynthesis is going to, forage yield is going to improve somewhat.
Especially if photosynthetic efficiency is the weak link in,
right,
in the chain here, of what's needed by the plant, all other things being equal. You know what I mean?
Exactly.
Mark your calendars for the 8th Annual Latino Farmer Conference on November 17th and 18th in Escondido, California. This annual Sustainable Agriculture Conference brings together Latino farmers, the agriculture industry, and advocates for sustainability. Join NCAT and NRCS for 2 days of workshops, local farm visits, networking, and learning, all in Spanish. Register today at latinofarmerconference.ncat.org.
And so when I was thinking about these 4 statements, it led me to think that to increase the food source for soil life, we need to manage plants in such a way as to maximize their root exudation. So then this caused me to do my own research. And so I can thank Google Scholar for helping me to find the journal articles for this. But I had to figure out what type of grazing management that would be needed to maximize root exudation. That's what I wanted to create the link, is like, what would grazing management look like in order to keep those root exudates at their highest level as much throughout the growing season as possible. So I came up with 2 questions. So the first question was, do plants release the same amount of these organic compounds all the time? And if they don't, how do they change? And then the second question is the amount of these organic compounds affected by how severely a plant is grazed? And if so, how do they change? So if I could get the answers to these 2 questions, we could make some inferences, right? We could make some potential linkages here, get some potential insight as far as, okay, without doing a full-on grazing management experiment to answer all of these questions, what's out there in the research that kind of gives us an indicator of what might be true. So I spent a lot of hours trying to find this stuff, and you can go into Google Scholar and you type in search string after search string and you look at hundreds of different articles. And so after a lot of hours of searching, I found 2 articles that shed some light on the matter. And the first one was an article that came out in 2001, where the researchers looked at 10 rice cultivars and found that the amount of organic carbon, so organic carbon is a signature compound that plant roots release, the amount of organic carbon given off by plant roots was lowest in the vegetative stage. It was highest in either the elongation or early reproductive growth phases. And that it was moderately high, something kind of a middle ground between the vegetative stage and the elongation and early reproductive stages. It was moderately high in that late reproductive growth phase. Now, this was looking at rice. Rice is an annual warm season grass. It's still a grass, but it does grow in a different part of the world than where a lot of our temperate pastures are at. But it was the strongest linkage. It was the closest thing I could find that would give me some idea of the rate of oxidation at different growth phases for the plant.
And in that article, there was some really good graphs kind of depicting this. And I am going to include a link to that publication in the show notes. So if anybody's interested, they can go and take a look at those, because it's really enlightening. Yes.
So how does this relate to increasing carbon dioxide so that plant leaves can take it up to increase photosynthesis? And so I was thinking, well, as the quantity of organic carbon being released in the soil increases, this gives soil life the energy to gather nutrients plants need and to multiply. And the greater the population of soil life that you're going to be having, the more active that they are, guess what? They're breathing more and they're respiring more CO2. So you can make a direct correlation between the exudation rate, the higher exudation rate in that elongation and early reproductive growth phase with a increase in CO2 being produced, because you're increasing the population, the activity of soil life. They're going to be respiring out more CO2, which then is going to be able to go back into the plant through the stomates on the underside of the plant leaf. So as the quantity of that organic carbon being released in the soil increases, so they're gonna that's just going to keep driving CO2 up. And this is really, I think, an important point to drive home, because then it ties into, well, at what stage should plants be when animals arrive? If we're trying to maximize root exudation, we're trying to maximize CO2 production to maximize photosynthesis in the plant so that we can get greater plant nutrient density, plant health, stuff like that. Does it matter when animals arrive? And if we're going to use this research to make some inferences, then the answer would be yes. And then that growth phase is really, you're looking at that elongation, early reproductive growth phase, if we're trying to, again, maximize the amount of CO2 being released. And as we talked about in, I think it was session 3 of our advanced grazing webinar, we talked about for the rumen ecosystem, whether we're talking cattle or sheep or any other ruminant that we may have, and we were talking about manure scoring and being able to judge how well the rumen is functioning. What's interesting is this lines up with really what is the best quality of forage for most animals, where they have enough effective fiber in the diet already, so that they're not having really runny stools, really runny manure coming out the back end. And it's really in that elongation, very early reproductive growth phase.
And whenever I saw this research, it hit home how everything works together. We've been talking about grazing at the elongation phase for a while now, because of the tighter ratio we have between protein and energy, for one thing.
Yes.
Right. And to see that it lines up with root exudation as well, just gives you a picture of this, of the complexity and design of this system. It's just amazing,
It is. I mean, I was just flabbergasted when I realized, from a plant standpoint, if we're trying to maximize photosynthesis, guess what? The growth phase that helps to maximize photosynthesis also happens to be the growth phase that is really going to provide the most balanced diet for livestock. So I'm like, I love it how those two actually line up. That's beautiful.
Oh, it is. So that was your first question. Then your second one basically was looking at like grazing severity and what you found in the data.
Right. So I did a bit. I went back and spent more hours in Google Scholar trying to dig. And then I came across this study that looked at the effect of grazing severity on root exudation for plants in an alpine meadow. And this was research, I think, that's been done within the last 10 or 12 years. And so they looked at 3 levels of grazing severity. So they looked at a no grazing control, because you always have to have a no grazing control whenever you do a grazing study. Then they had a moderate severity, and then they had a heavy or severe grazing. Okay, so we're just talking about how much of the plant we're allowing animals to take. So the research showed actually that heavy or severe grazing significantly decreased root exudation when the grazing was actually taking place. And it somewhat decreased root exudation after a 21-day recovery period. Now, that's somewhat of a decrease, but it wasn't a statistically significant decrease during the recovery period.
So that was like a decrease in exudation compared to the control, right?
Yes.
Okay.
Yes. So after a 21-day recovery period, the moderate grazing enhanced root exudation beyond the no graze control. So the control, the plant is just, it's not being harvested. It's just being left alone. So if it's not being defoliated somewhat, why would it be increased?
Yeah.
All of a
Yeah.
sudden decide to increase
Its root exudation is flat, basically.
Basically, yeah. For the no grazing control, it flatlines. And so then as I was looking at this, it's like, okay, well, that's interesting, because while the animals are in the paddock, if our goal is to, again, increase net photosynthesis, to turbocharge photosynthesis so that it's occurring at a higher rate, the amount that we allow animals to take does matter. Because if we do graze it severely, meaning taking it down very close to the ground surface, while the animals are there, the root exudation does significantly drop off on the plant, because you are really shocking that plant. Okay. Now in the recovery phase, it does put out a lot of root exudates, plants that are severely grazed, even when compared to moderately grazed ones. But it's all about, if I'm trying to maximize photosynthesis for as long a period of time, really, the time that the animals are there, if we choose a moderate level defoliation, and when I say that, this is kind of environmental dependent, context dependent, but if we're looking in that, somewhere in that 40 to 60% range, with what I would call a moderate level, severe or heavy grazing would be like, 80, 90%
defoliation. Yes.
Defoliation of what you're removing. So if we're trying to achieve maximum photosynthesis for as long a period of time, the time that the animals spend in the paddock is also part of that time, and a moderate level of grazing severity helps that root exudation to stay at its highest level. So that was the other insight, for me, as far as kind of attuning to our grazing management. Does it matter when the animals arrive? Yes. What the growth phase is of the plant. And then does it matter once the animals are there, how much do we allow them to take of the plant? Does that impact the amount of root exudation and eventually the amount of carbon dioxide that's being emitted by biology and going up through back into the plant leaf?
Right.
And the answer to that, is yes, according to the research, that I've been able to dig up. And I'm more than happy to look at, other research that shows other things, that gives us further insight. But this is what I was able to find by spending a considerable amount of time, trying to find the answers to some of these questions so that we can get some sort of a little bit of a science behind, why is it that we graze the way that we do, and if we're trying to maximize photosynthesis, like what John Kemp was talking about in a grazing context, how do we do that?
Yeah. Well, and what really excites me with this research as well is particularly looking at the grazing severity side. Again, just like the nutrient density in plants and having animals go in whenever the plants are at a higher stature and a little more mature makes a lot of sense, and that plays out in some of the things that we've read about. So does the grazing severity side, because that really dovetails nicely with the long-standing ideal of take half, leave half, right?
Right.
I mean, there's a reason we do that. We want to leave leaf area. We want to leave litter. We want to leave something there to capture rain and slow down that impact. We want to keep the temperature down. During hot summers, we want to maintain, water within the soil and things like this. Well, this moderate. And like you're saying, it could be anywhere between 40 to say 60% defoliation is really playing out with the amount of root exudation you get during those periods. So again, it's further substantiating some of the practices that we're doing. And it's given us a little more insight into what's happening whenever we do these practices.
Exactly. One of the benefits, and we touched upon this a little bit earlier in our conversation today, is that one statement that John Kemp said about, where's all this extra sugars if the plant increases photosynthetic rate from 15 to 20% to like 40% or 50 or 60%. And if you don't see all of that being resulting in a 2 or 3x increase in plant biomass, where's all that extra sugar going?
Mm-hmm.
And for those of us in a grazing context, most of us are familiar with the term BRIX, BRIX.
Right.
So that's just measuring the dissolved contents, the dissolved solids in the sap of the plant is basically what that is. A short way of saying is, they use bricks in the wine industry, in the juice industry to measure sugar content in the plant. So the higher the bricks level, the higher that number goes, the sweeter it is.
Mm-hmm.
And so when we look at the higher that bricks level, the more